G'day, good morning, good evening. Thanks for listening. This is a study sesh for a university course, Microbial Biotechnology. So I'm just going to go through a lot of questions, see how long my voice lasts, and that's going to be about it. So I'll go through some questions and then you'll have to read a lot, so I'm not going to read them all. So the whole, you want to get a bit of A4 foolscap and you put it on the screen and sort of get stuff around it to prop it up, and that stops this section. So you put it up to there so you can't see the number of what number it is, and you can't see where the dots were, because the answers there can really sway your thoughts. So that's how I do it anyway. And if you're going back through this, you'll probably want to use the pause button, because I'm going to go pretty quick because there's heaps of questions. What's the most widely used system of naming organisms? Binomial nomenclature. What are three domains proposed by Carl Woese? Bacteria, archaea, eukarya. Name one characteristic of aseptic hyphae in fungi. They have cross walls with pores for cytoplasmic streaming. What does the abbreviation PFGE stand for? Pulsed field gel electrophoresis. What does the binomial nomenclature system assign to each species? A genus and a species. What is the process of identification in taxonomy? Determining an organism's identity by comparing its characteristics with known organisms. What makes cyanobacteria unique among bacteria? Ability to perform photosynthesis using chlorophyll. What does serology study? Blood serum. What is the basis of phage typing in bacterial classification? Susceptibility to specific bacteriophages. What's the first step in DNA to DNA reassociation? It's going to be fragmented. What role does spike proteins play in some viruses? They help attach virus. They help the virus attach to and penetrate the host cell. What is the purpose of gram staining? To identify between the both. What's the primary function of the nucleus in endokaryotic cells? It's the DNA replication, transcription, and gene expression. What triggers the formation of endospores in bacteria? Nutrient deprivation. What's the main reason for recognizing archaea as a separate domain? Their genes and metabolic pathways being more closely related to eukaryotes. Which of the following component is the cell wall of fungi? It's chitin. What makes cyanobacteria unique among bacteria? Its ability to perform photosynthesis using chlorophyll. What types of enzymes do lysozymes contain? Acid hydrolyzers. What's the first step in bacterial binary fission? It is DNA replication. What are hormongia in cyanobacteria? The chains of cells involved in reproduction. Which of the following terms describe microbes that thrive in extreme conditions, like springs or salt lakes? Extremophiles. Which of the following pathways is favored by microbes at high temperatures due to its thermophilic adaptation advantages? It's the pentose phosphate pathway. Which feature enables Deinococcus to survive extreme gamma radiation? Its efficient DNA repair mechanisms. How does Deinococcus counteract reactive oxygen species induced by radiation? It accumulates manganese complexes that protect DNA repair enzymes. What pressure conditions are hyperpiezophils adapted to thriving? This is where Latin comes in handy. There's a few, if you can break it down in Latin, you can get the answer. I'm not going to give a blooming, but anyway, the answer is high pressure over 100 megapascals. Which of the following is not an adaptation strategy of thermophiles? A large genome size. Which of the following properties contributes to the thermophilicity of thermophilic enzymes? The compact hydrophobic core. The pH range of acidophiles and alkaphiles are what? What's their pH range? It's anyway, it's 5.5 and then 8 to 11.5. How does high pressure affect protein translocation in microbes? It destabilizes rhizomes and translocation factors. How does reducing water activity in food prevent microbial spoilage? It limits the growth. In translation, how are the codons read on that messenger mRNA? In the 5' to 3' direction. Which of the following is the correct order in the steps in transcription? So it starts with promoter, ends with termination. Which of the following is a structural gene found in the lac operon? It's LAC-Y. What does CAP stand for in the context of the lac operon regulation? It's the catabolic activator protein. What is the role of DNA polymerase in DNA replication? It adds nucleotides in the 5' to 3' direction, requiring 3' OH group. What's the role of DNA polymerase in DNA replication? It adds a nucleotide in 3' engine. What happens after sigma factor dissociates during transcription? The core RNA polymerase continues synthesis. Which of the following correctly describes the role of the LAC-Z? Encodes beta-galactolase, which hydrolyzes lactose into glucose and glucose and galactose. What are mRNA that contain numerous genes in a single transcript called? Polycistronic. What's the primary function of the single-stranded DNA binding SSB proteins in DNA replication? To prevent separate DNA strands from re-annealing. What organism grows on the erosin methylene blue agar with blue-green sheen? Coli. Which of the following methods of estimation may require preparation of tenfold serial dilution? Spread plate technique. Which of the following methods of microbial control uses moist heat? Autoclave does. Which of the hydrocarbons built from 5 carbon isoprene units called? What are they called? Terpenes. The luciferase luferin enzyme complex is derived from which of the following organisms? Botonius pyralis. Thiamine dimerization can be reversed through a process. Photoreactivation. What is orthophthalate prepared as a disinfectant over glutaraldehyde? Why is it different? It's all those reasons. Which of the following was the first amyloglyceride to be discovered? Streptomycin. Something is a method used to preserve and sterilize food by subjecting it to extremely high pressure. Pascalization. Something denotes the radiation dose caused a 10-fold reduction in the microbe population. Decimal reduction dose. Which of the following tests is used to screen bacteriophages? The plague assay. Which species of penicillin is most widely used for commercial production? It's Penicillin chrysogeneum. Which of the following editing tools can be used to develop a microbial cell factory? It's all of the above. Something involves direct contact between donor and recipient cell via a filamentous structure called sex pilus. Conjugation. Which virus first showed oncolosis in a lab setup? It's the VACV. Penicillin production requires the cells to be in which phase? It's the idiophase. Secondary metabolites are produced during which growth? It's the stationary growth phase. Which of the following is not a part? That was actually a question, the other one, food microbes. Which of the following is not a part of downstream processing? Media formulation. Which of the following group of enzymes aid in digestion? Starch. Amylases. Something is a computational approach used to predict the flow of metabolites through a cell's metabolic network. The flux balance analysis. What is the primary goal of classical biological control? To control pest populations. Which of the following is not a type of biocontrol agent? Herbicides like glyphosate. Which of the following fungi is used to control soil-borne pathogens like Fusarium and Rhizotonia? Trichoderma species. How do biocontrol biopesticides differ from conventional pesticides? They control pests through non-toxic mechanisms. Which of the following mechanisms promotes plant growth under stress conditions induced by climate change? Reduction of ethylene helps heaps in veg. How does the mold strain protect papaya plants from papaya ring spot virus? By activating the plant's defense mechanism. Which natural enemy is used in seasonal inoculative release of Mexican beetle? Pedioburus genus. Which of the following is the example of the botanical used as biochemical biopesticide? Neem. What are semiochemicals primarily used for? To communicate between plants and insects. What's the primary mechanism of Bt crops? They produce a protein that causes cell lysis of the grubs. You can read all this. Nearly done. 5 to go, but my voice is sort of sore a bit. So there you go. Oh, I can do that, can't I? So read through while I have a drink of my water. What are the bioprocesses? What's eutrophication? Excessive growth of algae. Which group of bacteria involved in oxidizing ammonia to nitrate? Nitrosomes. Which of the following is not a mechanism involved in microbial bioaccumulation? Bioleaching. What's the primary function of rotating discs in rotating biological control system, contractor system? To provide the surface area. What does membrane anaerobic reactors used to treat organic waste? Well, it's written in the title there, anaerobic digestion. What's the primary function of biogeochemical cycles? It's nutrients, recycling nutrients. Which enzymes primarily involved in microbial-induced phosphate precipitation? Phytase. What's the two mechanisms involved in autotropic bioleaching, direct and indirect? Which of the following processes is involved in natural attenuation for reducing environmental contaminants? Active microbial inoculation. Righto. My voice is sore. You can read this. Read your questions and press pause when you're ready, please. Next one. Last one. Week 12. There we go. That's it. I hope you got something out of this. I'm not going to go through them again. I myself going to go through this and more of the slides. We have a little bit of time. I'll show you some more slides. Some of history at the start, taxonomy, nomenclature. It's pretty cool. It's just strapped up. There's a chain staphylos group or cluster. Rod-shaped. So pseudomonas can be rod-shaped too. Oh, sorry, can be. They are. Corynebacterium. I need one of those species for CT. Spiral bacteria shapes, yes. Spirochetes, very good. One's tighter than the other. Other morphs, filamentous, stalked, and cleviomorphic. Filamentous, yes. Stalked. This is Paleobacteria, such as Acetobacter, exhibit the ability to alter their shape and structure and repossession changes. That'd be good under the microscope. Similar, sounds like amoeba. Classification of bacteria based on their flagella. Do you know plants have got a gene, a receptor, FLG22, that acknowledge this flagella? Classification of bacteria, brain straining. What's the difference in the cell walls? Positive. Modes of metabolization. Heteros also as chemodes and photos. Heteros, all those, yeah. Chemodes. Photos. Very good. Yes. Do not change that. Yeah, this is good. This is pretty common. This nitrosomus converts ammonium or ammonia into nitrite, which is what fish pee out. And then Nitrobacter converts nitrite to nitrate, which is the plant recognizable form, just like ammonia is. So lightning produces this. And then if you've got nitrosomes around, bang, with Nitrobacter, you've got your action. You've got free feed for plants. Why am I talking about that? This is a study session. Classification of bacteria seed based on cell wall characteristics. It was one of those questions on DNA denaturing. 16 arrows. Protein bacteria. Protein eating first. Bacteria. Streptolysis. Microbes. Cyanobacteria, they're cool. Yeah. Marine cyanobacteria. I'd batch culture this. Chlamydia. Bacteria in the phylum. Yes, for medical, okay. Spirochetes. I'll do this for another 3 minutes. A few examples of bacteria genre. With identification problems using the 16S. That's how they identify the bacteria with the 16S RNA. So identification problems. This is a while ago, 2007, yeah, because some of them are commercially available now. Domain, okay. What's the difference? Bacteria. Complex. Okay, considered to be among the most primitive life forms, resembling early cells formed 4 billion years ago. They're still around today. How can they survive? Alphas, yes. Alphas. Fungi. Septate, hyphate, and non-septate. Oh, okay. Multicellular forms, which may be septate or non-septate. Cell wall divide lines. unicellular. Bacterial. Glomus aclospora. That's what I need in my diet. Aclospora down the bottom. I've got glomus. Rosipus. It's one of the exam questions. We'll be up for a bit longer. Septate hyphae with simple septal pores in ascomatose. So interesting, all this stuff. Look at that, 1904. Cool diagram. Protists. Algae and protozoa. Protozoa. It looks so scary under the electron scanner microscope. I wonder if we've got any photos of it. Plasmodium. Coccus curleos. Worship. How cool is that? Remediate, it changes color based on the metabolic reactions after 24 hours. What I call taxonomy waste, so I morphogenically identify them. Oxygen experiment. There's a serology, study blood serum. Another question. Just typing. Suppose. Classification. Righto. I think that's about it. Yes. Thank you for having the study session. I hope it helps in your exams too, or just for your general knowledge. If you have any questions, you're welcome to put them below and I will answer them. Thank you again, and I hope to see you someday in my life. I do weekly. Thank you. If you did like this, if you got anything good out of it, press the like button, please. Trying to grow the family in agriculture, the nice family that it is. That's what the word, not family, the nice community. Yes, thank you. Good luck. Bye-bye. Thank you. Hello, Genetic Memory Farms. Sorry, I didn't say anything earlier, mate.